Hi, I'm Carly Moss, a second-year PhD student at the University of Maryland College Park, working with Sujay Kaushal. Today, I'll be giving an introduction to my research on the attenuation of different chemical cocktails longitudinally along Bull Run. As you saw in the previous presentations by Gabrielle, Shantanu, Caitlin, and Stan, they are researching and modeling the sources and concentrations of ions which are entering uosa. In my research, I am addressing the chemical retention, release, and mobilization which occurs in Bull Run because of uosa's effluent. So I'm focusing on that red box highlighted here. Currently, salt ions are not intentionally removed during the wastewater treatment plant process and could be contributing to freshwater salinization in Bull Run as well as the Occoquan Reservoir. Overall, I want to know if Bull Run can naturally attenuate the ions, metals, and nutrients that are added from uosa's effluent as it moves longitudinally downstream. To answer our question about what makes it downstream, we'll use the chemical cocktail approach and look at novel elemental combinations and signatures due to urbanization. So instead of looking at one element, we will look at a suite of ions, metals, nutrients, and organic matter. These cocktails travel together based on their chemical properties. We will be looking at the retention, release, and transformation of chemical mixtures longitudinally in Bull Run. Bull Run is influenced by urban, suburban, and forested land use, as well as a continuous input from the uosa effluent, which creates unique and new chemical cocktails with the potential to travel to the reservoir. During this research project, I'd like to address two questions. How are chemical cocktails transformed, retained, and released longitudinally from wastewater effluent? And how do shifts in land use influence the water quality? To conduct this research, teams from University of Maryland and Virginia Tech hiked along the Bull Run Trail and collected grab samples as well as field measurements. Each sample collected was run for ions, metals, nutrients, and organic matter. I'll be presenting two graphs comparing specific conductance and distance downstream in January and September. Previous work has shown there's a relationship between specific conductance and salt ions. On the x-axis, we see distance downstream with 0 meters at the uosa effluent. I'm presenting data from the main stem, which is in blue, and the tributaries in orange. We see there's a low specific conductivity before uosa, a spike of specific conductivity above 600 microsiemens per centimeter at uosa, which indicates there's a release of salt ions. Then we notice a rapid exponential decline in specific conductivity, as well as continual attenuation downstream. The concentration in the tributaries exponentially decays as we move downstream and impacts the specific conductivity of Bull Run, causing dilution. In September, we observed similar trends to the January data. There is a lower specific conductance before uosa, with a drastic increase of conductance above 800 microsiemens per centimeter at uosa, then a rapid decline in the concentration between 0 and 2,000 meters, and then attenuation. We also see that there is dilution from tributaries as well. One difference from the September and January data is the September data shows an increase in specific conductance as we approach the reservoir and the Bull Run marina. My next step is to use multivariate statistics to compare chemical cocktails of ions, nutrients, and organic matter from winter and summer sampling seasons to understand the sources and processes of retention, release, and mobilization of chemical cocktails in Bull Run. We have seen similar trends of attenuation along Bull Run in specific conductance for both the winter and the summer. There is attenuation of ion-specific conductance and nutrients in the winter along the flow path as well. Next, we want to address how chemical cocktails are retained, released, and transformed along Bull Run to better understand what enters the Occoquan Reservoir and potentially our drinking water treatment plants. Thank you.